that you're giving this talk. I thought it was one of the highlights of last year's Levine Symposium. I was so impressed.
Oh, thank you.
Well, I
Well,
just love learning things that I had no idea about. It was just a whole new vista of knowledge. It was terrific.
well, it was very exciting for us, and I have something completely different and new to talk about towards the end of the talk today, and hopefully you'll get your sense of whether it's something worth pursuing or not.
Well, it's a fantastic privilege and honor to welcome Tony Ferrante here today. Tony is an endowed professor at Columbia University Medical Center, I think as most of you know, and he's chief of preventive medicine and nutrition. He obtained the BA degree in physics at Yale, and both the MD and PhD degrees from Albert Einstein School of Medicine. And then he completed his residency and fellowship training and clinical training at the Diabetes Center at Columbia as well. He has a raft of awards. He's an exciting scientist and named lectures and so forth. But he's also done, I think, what I feel is something that is very rare, and that is he's made a tremendous, no, I'm going to use the word terrific, impact in the field, an unusual impact. And I think many of you know his classic paper in 2003 in terms of the JCI, where showing that obesity drives infiltration of immune cells, really created its own field, immunometabolism. And now hundreds and thousands, really, scientists and labs are working in this field that Tony's beautiful work created. That paper, by the way, has 10,000 citations. So if you're looking for real impact in a field, you know, that is a classic. And he's continued to, of course, lead the field. He's going to tell us more about what he's been doing more recently, and I think most of it's going to be unpublished. So again, we really appreciate that. And I would sort of end my introduction by saying, of all the scientists I know, and I know Tony, I think, quite well. We've seen each other now in many occasions, even on the tennis court. He's one of the most thoughtful scientists and originators and original scientists, you know, that I know in the field. So it's just a real pleasure having him here, and well, I guess in quotes, having him here and seeing him today. We really welcome you, Tony, and thank you very much for coming in and having this presentation. We really appreciate that. And we'll leave the questions till the end. And when you want to ask a question, just on the chat, just send me a note saying question. You don't have to write the question out, and then we'll call on you to ask the question, and Tony will be happy to answer those questions. So with that, Tony, again, thank you, and we're looking forward to your talk.
Mike, thank you. That was very overly generous. And I guess just before I start, I just want to say how, you know, sort of lucky I feel to, you know, first of all, to come to University of Massachusetts, where I have many friends and colleagues, but just also take a moment to recognize how, you know, lucky I and most of us are to be able to continue to do our work during these very difficult times. So I really appreciate the opportunity to tell you about the work that we're doing and recognize that it really is a privilege. So thank you. So what I'm going to talk about today is review some our model of how we think about adipose tissue, and in particular how we think immune cells play a role in normal lipid homeostasis within adipose tissue. So I'm going to review for the first quarter or so of the talk data and sort of a model of how we think about adipose tissue, and then talk about two quite different bits of research that we've been doing recently. One about the ontogeny of cells, and the other about promiscuous gene expression. So what I'm starting off talking with, or what I'm going to be focused on today, are the cells shown filled with lipid here, as indicated by red. And these are adipose tissue macrophages. They're a relatively unique population of cells within adipose tissue. Some of them, as you can see here, are multinucleated. and morphogenically are quite distinct. And as you'll see from my talk, they're heterogeneous in as a population. So just quickly to review the people who actually did the work, because of course I spend most of my time sitting in my office and then bugging people about experiments that they're doing, and just really go out and try to get money so that they can do it. But this is work that I'm going to sort of summarize that's been done over the period of about 15 years by actually all graduate students from my lab. And then the most recent work that I'm going to show you is work that's done by a recent graduate student who just graduated, Ethan Edwin, and a longtime colleague of mine in the lab, Xiaoyuan Xu. So that's Ethan, and that's Xiaoyuan. And as my son criticized me for my being cheap on my webpage, it looks like all the photos that I've taken of people are prisoners, but I assure you that they are able to leave, as Ethan has shown in terms of graduating. So I'd like to start with sort of what the paradigm is that sort of emerged over the last decade and a half about our thinking about immune cells in adipose tissue, and in particular macrophages, and the sort of dichotomous idea about these cells being either bad guys or good guys. So there is an idea that they took a polarity in sort of using classic terms, M1, M2, along the lines of Th1, Th2, and that their function, their effect on adipose tissue was largely measured through their maladaptive effects, and that this was viewed again through a prism in which the cells were seen as M1 or M2, where M1 are pro-inflammatory and M2 anti-inflammatory. And that grew out of, I think, some classic experiments that were done a decade or more before even we were aware of adipose tissue macrophages in Bruce Spiegelman's lab by Gökhan Hatemислagil. And his work, what he showed was that TNF alpha is expressed in obese adipose tissue. And for folks who are not old scientists like me, this is a northern blot, a way we used to measure RNA. And what you can see here in alternating lean and obese samples of adipose tissue, TNF alpha is expressed by adipose tissue of obese animals. And it had long been known that inflammation induces insulin resistance. So very reasonably, Gökhan and Bruce postulated that TNF alpha contributed to insulin resistance. And so this is the experiment that implicated TNF alpha in the development of obesity-induced insulin resistance from their science paper in '93. And essentially, this is a sort of a modified glucose clamp where there's infusion of hyperinsulinemia is induced, and then glucose is infused to keep the animals insulin sensitive. And what they did, and what's shown in the black squares, they infused TNF alpha receptor to antagonize TNF alpha, and showed that each time they did that, a few minutes later, the mice required more glucose. So arguing that TNF alpha was inducing insulin resistance. So again, that was the background to studies and looking at immune cells in adipose tissue, and sort of setting up the field to view them as sort of bad guys. So this is what I'm going to review, basically the 17 years of work from not only our lab, but many labs in trying to understand the role of these immune cells as well as other cells in adipose tissue. I'm going to really just focus on the macrophages, but be mindful, as you'll see later in the talk, that there are many other types of cells in adipose tissue that are very dynamic and changed by metabolic state. So I'll just sort of review and remind you that macrophages throughout the body actually have roles that are adaptive and tissue specific. We'll review some of the nomenclature because I realized that can sound like a little bit of gobbledygook, particularly when immunologists are talking, and I'm not an immunologist, about IL-347 and CCR98. to just give you some key highlights about some of the nomenclature that we use, talk about sort of this ATMs as the bad guys, and then present some of the new data that's unpublished, sort of the ontogeny of macrophages in adipose tissue, different subpopulations, and present to you our working model for what we think adipose tissue macrophages are doing to help contribute to lipid homeostasis. And then finally, talk to you about a discovery that actually multiple people in the lab made independently and that we're still trying to sort of figure out its relevance, and that's promiscuous gene expression by ATMs. Okay, so just a reminder that while macrophages were described more than a century ago by Ilya Mechnikov as cells that were critical in fighting pathogens and invading microbes, he also argued in his initial papers that they help to restore harmony and keep harmony in tissues. We'd probably call that homeostasis now. And that's been borne out, particularly over the last 2 decades or so, where it's become clear that macrophages serve not only sentinel functions and direct immunologic functions to deal with pathogens and injury, but also play a role in maintaining normal tissue function. So in the CNS, microglia are critical for pruning. Perhaps best studied are the osteoclast macrophages in bone that are critical for bone resorption. More recently, work has shown that cardiac macrophages play a critical role in AV node conduction. And similar to, I think, to what the role of macrophages in adipose tissue, alveolar macrophages play an important role in surfactant clearance and a lipid cycle there. So I think, again, the best understood and best studied of the tissue-specific functions of macrophage, the trophic functions, are of osteoclasts. So osteoclasts, like adipose tissue macrophages, are multinucleated macrophage cells. They are derived from progenitors that are regulated by CSF1, colony-stimulating factor 1. And they seem to work in direct opposition to osteoblasts. So osteoblasts are the bone-forming cells, so they make mineralized bone, and osteoclasts, of course, degrade this. And it would seem to be just a useless futile cycle of deposition of bone and degradation of bone. But we know that actually this is important to maintain healthy bone. And I'm going to argue that similarly in adipose tissue, a recycling of lipid is important to maintain healthy adipose tissue. Here we know that if you disturb the balance, either in favor of osteoblasts or osteoclasts, then you can end up with osteoporosis or osteopetrosis. But even that this basic bone resorption cycle is important to lay down fresh, healthy bone. Okay, so just some quick nomenclature. So we throw around the marker F480, which is a marker of most macrophages, particularly in mice, and I will use that often. CCR2 is a chemokine receptor that is critical for recruitment of macrophages or precursors into adipose tissue, but also precursors to many other, of monocytes to many other organs. And then two antigens that integrins actually, that are used as markers for FACS analysis, CD11C and CD11B. And together, we often refer to cells that express macrophages in adipose tissue that express a combination of these antigens as FB, meaning that they express F480 and CD11B, but not CD11C, versus FBC that express all three. Okay, so again, the story that, or the setting for which most common paradigm that's promulgated these days is that there's the macrophages become in adipose tissue are confused or for some reason upset by their environment, get recruited and activate an inflammatory response. and that this leads directly to their contribution to dysfunction of adipose tissue. And this, I'm sorry. So what we wanted to ask, though, are what functions do ATMs serve? Is it really that they're just confused immune cells that end up in the adipose tissue, thinking that there are pathogens there, but not really? And are they really good and bad ATMs, or can we perhaps discern differences in function? So Xiaoyuan Xu started some of these studies with Ethan, and her original studies started back more than a decade ago, trying to parse out differences in adipose tissue macrophages in lean and obese animals. And this is work, again, that's been published a long time, but actually I think Mike's lab found, and other labs certainly have found something very similar. So if you look at, for example, TNF-alpha expression, consistent with what GoComp found almost 30 years ago now, there is an increase in overall expression, both of the gene as well as the protein, TNF-alpha. And so it can be as much as 10 or 15-fold. But if you look at individual cells, so this is an Xiaoyuan FACS-sorted F480 positive so all the macrophages in adipose tissue, and the two flavors of macrophages that we were aware of back then, the FB and the FBC, and you look at TNF-alpha expression per cell, there really wasn't much difference at all. And this is at the RNA level, but similarly, if you look at the protein level, there really is not much difference between ATMs from lean or obese animals. The other thing I'll point out in passing is that the number of mice that she needed to sacrifice to get a detectable amount of protein by this Western blot was on the order of 10 to, or actually in the lean animals was something like 10 to 15 animals per lane, 4 to 5 in the obese animals. So there doesn't appear to be certainly for TNF-alpha, a switch in sort of the inflammatory profile. And indeed, when she did both qPCR as well as at the time microarray data expression analysis, you can see that for the genes that are classically associated with inflammatory phenotypes, there wasn't an induction, a wholesale induction of some acute M1 inflammatory transcriptional program. And so comparing the lean in white and the obese ATMs in black, and it didn't matter which subclass she looked at, she again saw the same thing, that there was an inconsistent alteration, and certainly not an alteration towards an inflammatory phenotype per se. Now you can pick one or two genes and in fact find increases in expression, but typically the increases in expression were relatively modest, certainly compared to the typical infection, but also again was not consistent across what one would see, for example, in an infection. So, but there are differences in ATMs from lean and obese animal, and this is, I think, the point that I'd like to drive home, is that when you look at the transcriptional program, the thing that jumps out at you is that the programs related to lipid metabolism are dramatically altered. And then when you just look at the cells, the thing that becomes apparent, as illustrated here in this live confocal microscopy, is that these macrophages, indicated here by the F480 staining, are chock full of lipid, or many of them are, and this is from an obese animal. And in addition, another program that Xiaoyuan saw that was upregulated was a lysosomal or degradative program, catabolic program for lipids. And indeed, when she looked for lysosomes at using many different techniques, but indicated here by lysotracker or fluorescent dye, you can see there's a large number of acid-filled compartments within these mature adipose tissue macrophages. So one of the things, the thing that was clearly different between lean and obese ATMs was the accumulation of lipid, as well as the turning on of this degradative program or catabolic program for lipid degradation. So putting together a whole lot of data collected, again, not only from our lab, but from other labs as well, what I think has emerged is that when that ideally, healthy adipose tissue keeps the lipid within the adipocyte, but that the immune cells play a role in trying to both buffer when excess lipid falls out of an adipocyte, or as I'll sort of indicate here, when there's a lipid cycle per se. So we showed again about a decade ago that if you induce lipolysis either through fasting or through weight loss or through pharmacologic means, you get a release of obviously of free fatty acid, and this causes a very rapid recruitment of precursors and the development of macrophage population very rapidly in adipose tissue. What also happens, and happens more slowly, the accumulation of macrophages in obesity, however, appears to be driven by something that we sort of stumbled upon and published last year, and that is the production of these lipid-filled vesicles, which appear by most criteria to be exosomes. And these triglyceride-rich vesicles are then the source of the lipid that I showed you in the confocal image a couple of slides ago that accumulate in the macrophages. And a series of students in the lab, as well as Xiaoyuan, show that the lipid in these cells are shuttled to lysosomes. They then, it is degraded into free fatty acids. It also causes, data I'm not showing, but was published a couple of years ago, so the free fatty acids are released, but it also leads to the production of anabolic factors, factors that would encourage adipocytes to be happy, but also to store more lipid. So this would suggest that there is a lipid cycle, that the adipocyte releases lipids in this relatively energy efficient way, not requiring a whole lot of energy, in these triglyceride-rich particles. They're taken up by macrophages. This process increases dramatically when animals are obese, leading to more macrophages being recruited. The lipid is hydrolyzed through a lysosomal-dependent pathway that's independent of autophagy, and the free fatty acids themselves are then taken up by the adipocytes and reesterified. And if you block this as what occurs in the absence of lysosomal acid lipase, either in mice or in rare humans, what happens is that while those mice or people are born with normal amounts of fat, over time, the fat atrophies. And not because of dysfunction of the adipocytes themselves, but because this lipid cycle is broken. So in the case of mice, they're born with normal amounts of fat at 5 and 6 weeks of age. They have normal amounts of fat, but by 24 weeks of age, most of the fat is actually gone and actually ends up being accumulated in other tissues. Okay. So now I want to tell you, so that's sort of the framework in which we try to understand the immune cells, in particular macrophages in adipose tissue. And but we want to figure out a little bit more about the function of these cells. And one of the things that was quite apparent from years ago, that there is not one uniform population of adipose tissue macrophages, but that there are several, as indicated, as I told you, by the expression of antigens, let's say CD11C and CD11B. And in order to understand the functions more fully, we thought that we would need to really identify the subpopulations of macrophages, with the idea that that different flavors of macrophages may in fact have different functions. So this led to just some basic questions. Where do they come from? How many different types of macrophages can we identify? And do these different flavors, at least based on either their origin or some of the proteins that they express, do they possess different functions? And in particular, based on our model, are they subservient to some lipid functions? Or in fact, are they regulated by non-lipid functions? So we decided, or Ethan decided, to approach this from looking at macrophage ontogeny. And so just a brief diversion to remind you that, again, over the last decade or so, it's become apparent, contrary to the sort of long-term dogma, that tissue macrophages are derived from two different, essentially two different pools, certainly in mice, and I think most of the data is consistent in humans as well. And so those are the prenatally derived ones and the postnatally derived. That is that the precursors for some adipose tissue, for some tissue macrophages are derived either from the yolk sac or from the fetal liver, while others are derived postnatally from circulating monocytes, which ultimately are derived from bone marrow. And it's become clear that tissues are different in their relative composition of these sorts of macrophages. And this became perhaps most dramatically demonstrated in the brain, where nearly all of the microglia, the brain macrophages, are yolk sac derived. This was very nicely shown by Miriam Marab at Mount Sinai and her collaborators. And then, at the other extreme, if you look in adult mice, and we think the same thing from transplant studies in humans, that the gut-derived macrophages are nearly all derived from circulating monocytes, bone marrow, and turn over very rapidly. And then other organs in between have different combinations of cells that are either derived from yolk sac or fetal liver macrophages that are populate the tissue pre-birth versus those that are in various combinations derived from circulating monocytes. So Ethan, when he started out, asked a question which was unknown, was what is the ontogeny of adipose tissue macrophages? And is there evidence that they, in fact, there may be ontologically different macrophages? So we had, many years ago published that CCR2 and blocking CCR2 could reduce the number of macrophages in adipose tissue in a high-fat feeding model. And so that suggested that at least some of the macrophages were derived from circulating monocytes, again, consistent with what would be considered the classic paradigm. Actually, work done not too far from where you guys are sitting in Mike's lab and Miriam's lab looked at and demonstrated, I think for the first time, that macrophages actually could proliferate locally within adipose tissue. So this gave us a hint, certainly, that there may be multiple populations that have different precursors, some that arise from circulation, others that may be arising from local precursors. So, and I think that there's been conflicting data along these lines, and certainly there's no, I think, definitive way of a single experiment that can show this. So in any event, just to remind you again, F480 is a marker of macrophages, but in many other tissues, a number of tissues in liver, in the CNS, relatively high expression of F480 is associated with resident precursors, so cells that are not derived from monocytes or bone marrow, whereas the expression of CCR2, that chemokine receptor important in recruitment of many macrophage populations, is actually high in many of the recruited or bone marrow derived cells. So with that, Ethan went and took a look at some of his FACS data to see if he could discern populations based on that, and then plan to do a series of different studies, each independent, to determine whether there were in fact ontologically distinct adipose tissue macrophage populations. One is MIB dependent, so a genetic technique of ablation, parabiosis experiment, which is truly just a surgical technique, and then a fate mapping experiment using a tamoxifen inducible CRE. Okay, so first he went and looked back and said, okay, can I see in fact differences between F480 populations if I look more carefully at FACS plots of ATMs, can I distinguish a low from a high? And in fact, he could pretty easily. So this is data from a lean animal, but he also sees something quite similar in obese animals. So this then provided him with a hypothesis that, consistent with what's seen in some other tissues, that these F480 low macrophages might be enriched for cells that are recruited from monocyte-derived precursors, and that the F480 high ATMs might be derived from resident precursors. So he set out on doing these two, with this hypothesis, using these three different techniques. So this first technique, which is, again, was developed in the last decade or so, takes advantage of the fact that the MYB transcription factor is required for definitive hematopoiesis, and deleting it leads to bone marrow failure, and then mice die. Now, the advantage of doing using this technique to ablate bone marrow is that, unlike radiation techniques, you are not affecting resident cells that have left the bone marrow that may reside, for example, in the brain, that are not dependent on MYB. So what Ethan did in this experiment was take these MYB flox mice that are will be in Cree can be induced by poly-IC, a viral mimetic, and that leads to MYB deletion. So what this happens then, what happens in these mice is that their bone marrow, to varying degrees, fails, and what he did to rescue them was to inject GFP MYB positive bone marrow cells from a donor mice. And so what this leads to is that you get rid of the bone marrow from the recipient mice, and that all the bone marrow, or bone marrow that engrafts, is now identifiable by expressing GFP. And when he did this, his hypothesis was that the bone marrow derived F480 low ATMs would be green pretty quickly, and that the remaining F480 high would not. And that's in fact what he saw. So he's looking here at T cells, again, which are not bone marrow dependent. And so their fraction of those cells in adipose tissue that are derived from the bone marrow and are green is very low. In contrast, the F480 low ATMs, both at 14 days and 84 days, the vast majority of them are in fact green, indicating that they came from bone marrow precursors. Consistent with his hypothesis, the vast majority of the F480 high ATMs, though certainly not all of them, were GFP negative, indicating that they were residing in the adipose tissue or derived from cells that are not bone marrow dependent, and therefore from precursors that were residing in the adipose tissue per se. So that was one technique. There are all sorts, caveats that can arise from doing this approach. So Ethan decided to take a second approach, and that was to use parabiosis. So here you basically surgically create Siamese mice, and again, using a green mouse and a non-green mouse, he was able to track movement of cells. So when you create these parabiotes, they establish a shared circulation. Really, a very small fraction of the circulation is shared, something on the order of about 1 to 2%. But nonetheless, it does allow you to track cells, immune cells that travel by the circulation between the mice. And when he did that, he found essentially the same exact result. So looking 28 days post-surgery, you can see that very few of the F480 high cells, the ones that he predicted would be arise from precursors in the adipose tissue, were in fact derived from the parabont pair, whereas the majority of the cells were actually that were F480 low were derived from the periont. Now, you may say, oh, 30%. But keep in mind that 50% would be equal sharing. The circulating monocytes actually achieved a steady state of about 30%, meaning that 30% of the monocytes in any one mouse are derived from its parabiote partner. And that has to do with the kinetics of turnover of monocytes. So essentially, almost all of the F480 low cells in the adipose tissue are derived from circulating precursors. But again, it's not 100%, and certainly it's not 100% for the F480 high. And then finally, his last experiment to try to nail this down was to do a fate mapping experiment in which he took mice that carried a tomato allele that becomes expressed and therefore marks the cells in a lineage-dependent fashion under a Cre-dependent fashion, and this was driven by the CSF1 receptor promoter, and it's tamoxifen dependent. And CSF1 receptor is really limited to monocytes, macrophage, dendritic cells, and very few other cells, trophoblastic cells, and maybe a few other cells in the testes. So it's very good for marking, certainly, macrophage lineage cells. And again, he did this experiment. What he saw was, for monocytes, he saw what was expected. About half the cells, or slightly less than half the cells, about 40% of the cells are actually labeled when you do a single injection of tamoxifen the way you did. And then over the next several months, actually within a couple of weeks, almost all the monocytes are no longer labeled, and that's consistent with our understanding of the turnover of monocytes. So he then looked in adipose tissue, and consistent with the previous two approaches, he saw that the F480 high largely remained labeled green, or rather red, in this case tomato. So they started out after 3 days, 40% efficiency of being labeled. At 3 weeks and all the way up to 7 weeks, the majority of the cells still remained labeled, meaning that they were not replaced by circulating cells. This was not the case for the F480 lows, where by 3 weeks, the vast majority of them have already been replaced. Again, consistent with the other data. So his hypothesis was correct that the F480 high ATMs are largely derived from tissue precursors, but probably represents a heterogeneous population. And that similarly, the F480 low are largely, and probably even a little bit more homogeneous, derived from bone marrow circulating precursors. He was also able to, based on all these data, sort of give an estimate to the half-life of these various populations. So the 3 different techniques generated half-lives that were slightly different, but all, within the same ballpark. So the F480 lows have half-life that between a week and 2.5 weeks, based on the bone marrow transplant MIB and the fate mapping experiments. And then by comparison, 2 order of magnitude longer lived are the F480 high. And the turnover rates were pretty similar between different depots, so perigonadal versus subcutaneous versus retroperitoneal, all had similar, again, at least an order of magnitude or different in terms of their turnover rates. So it was clear, as we suspected, that none of these populations would be homogeneous, and I think it was clear based on the fact that the F480 high population appeared to be some that were derived from tissue, or the majority were derived from tissue precursors, but there's also seemed to be some that were derived from circulating monocytes. So he did bulk RNA sequencing, and consistent with what he predicted and what we knew, the resident ones, so the F480 high, express high levels of CSF1, the growth factor for macrophages, and relatively low expression of CCR2, although detectable nonetheless. And by comparison, the converse was true of recruited ATMs. What was useful, and the reason partly we did this experiment, was we were trying to identify different factors that might be useful in regulating these cells so that we can assess their function. CX3CR1, which is expressed by both but relatively low, certainly didn't distinguish them. Again, based just on bulk sequencing, these two F-ready populations were clearly very distinct when you did PCA plot with almost all of the difference attributable transcriptionally, at least, to this one, the PC1 axis. Okay, so that led to sort of just a broad sense that the resident macrophages were F-ready high, they express low CCR2, and their half-life is at least 3 months. In contrast, the recruited ATMs were CCR2 high expressors, low expressors of F-ready, and had half-lives that were less than 3 weeks. So we then wanted to look to see if we could, discern, different subpopulations with the goal of trying to identify different functions, believing that different subpopulations would subserve different functions. So this is just really a very simple, straightforward single-cell RNA-seq experiment that Ethan did, and he did it from multiple mice so that we were hopefully not led astray by conditions of any one mouse or any one day of experiments. And so looking at more than 12,000 cells, he could identify many different immune cell populations. The good thing is that the vast majority were in fact immune cells. There were some pre-adipocytes that sort of slipped through, but more than 99.9% of the cells were in fact immune cells. What you can see here, and this is from lean mice, is that the adipose tissue macrophages, are the ones that he identified as macrophages, represent the lion's share of cells, but certainly not all. The next largest population of cells were T cells, and that there were other substantial populations of cells, all of which changed pretty dramatically, actually, with the onset of obesity. I'm not going to sort of talk about that now. But in particular, NK cells, some dendritic cells, and much smaller population, of B cells. And among the macrophage populations, there were 9, but there were 6 that were sort of major populations that were distinguishable based on their transcriptional profiles and aligned seemingly, we thought, with potential functions. When he also confirmed, based on the transcription data, what he had seen on his ontogeny studies, and in particular, that F480 was expressed in a gradient across the cells. So you can see here on the left, there the intense. So these are UMass plots. And what you can see here on the left in intense blue are the cells that express F480 high, but all these other macrophages shown here express some degree of F480. Here on the right is shown in red CCR2 expression and CSF1 receptor expression. So we had known, and it's been published previously, that CCR2 is also expressed in T cell populations and and as well as in NK T cells, as shown here. But the vast majority of the other cells that express CCR2 are in fact macrophages and are distinct from the F480 high macrophages. And that is to say that the CCR2 expressing cells were largely the F480 low cells. And similarly, and sort of strikingly, there was really a dichotomous expression in CSF1 receptor, the receptor for CSF1. Again, all of these cells in the macrophage lineage actually expressed detectable CSF1. It was actually an order of magnitude higher in the F480 high expressing the resident cells. The other thing that struck or stuck out to us was that these cells express high levels or were responsible for expressing IGF-1. And so, and these recruited cells seem to be marked by extremely high expression of LPL, lipoprotein lipase. So this suggested to us that these cells may have distinct functions where the resident-derived F480 high cells might be producing IGF-1 in direct communication with the adipocytes and trying to keep them healthy, alive, and storing fat, while the recruited ones may be more sort of on a mission like the fire squad trying to deal with excess lipid that might be floating through the adipose tissue. Unfortunately, these hypotheses have not been proved correct, at least tractable. So we've made various knockouts, caused expansion. Ethan tried expanding, doing different techniques to cause expansion in a specific way of these different populations, but could not. So we should have read Mike's paper a little more closely, and we would have realized that local overexpression of MCP1, the ligand for CCR2, would not only cause recruitment of the bone marrow circulating precursors, but actually also lead to proliferation of the local resident macrophages. So I wish I could tell you that and then we did some knockouts with IGF-1. We're going to pursue some other trophic factors that we think in combination with IGF-1 might be critical to maintain adipose tissue or adipocyte health and differentiation. But we have not, I don't feel like we've sort of come with a definitive answer to this. So we've identified the populations, we've identified them ontologically, and we're still trying to nail some functions to them. But what Ethan did do was to try to compare these populations to other cells from atlases that have been generated from single cell sequencing to see if he can get some clues. And so he used a mouse atlas which contained more than 900 cells or cell populations, and compared them to his the various ATMs that he identified using his single cell sequencing. And then he looked both at lean, obese, adipose tissue macrophages from mice that were on a high-fat diet for 2 and 8 weeks. And this is what he found. So he then compared and tried to see which of these myeloid populations that have been characterized previously these ATMs might be most similar to, in the hopes that that would provide further clues as to what these cells might be doing. So this is the dendrogram for that he did, and down here outlined in this box are the ATMs from his single cell sequencing. And what you can see, I guess not unexpectedly, when all the ATMs are put in this sort of compared to other populations of macrophages, they segregate to each other, so or are clustered with each other. Perhaps not surprising that they are most similar to each other, whether they be recruited or resident. But the other thing that was clear from this larger cluster is that all of these, or almost all of these other cells, are located in tissues where there's a large amount of lipid. So in, for example, involuting mammary tissue and lactating mammary tissue, as well as, for example, in the alveolar space where, again, clearance of surfactant is critical. So we think, again, that this provides some indirect support that these cells are involved in, or one aspect of their function is similar to other macrophage populations important in lipid metabolism and homeostasis. Okay, so this is, again, our working model. unfortunately, we haven't been able to, at least at this point, disable the function of any of the subpopulations of these macrophages. We're working diligently, or I should say the people in my lab are working diligently and trying to do that. And so hopefully in the next couple of years, we'll have a much clearer picture of specific functions of these macrophages. So something else emerged from these data that I just want to briefly tell you about. And it actually emerged from data both Xiaoyuan and Ethan did, looking at very different populations of mice for completely different reasons. Ethan's, as did lots of sequencing, as I mentioned throughout the talk, but Xiaoyuan also did sequencing for other reasons. And then we also went back and looked at some other data sets that we had collected over the years. So let me just show you an interesting comparison from Xiaoyuan's data, where she was comparing some ATM populations from lean and OB animals, leptin deficient animals. When she did some transcriptional differentiation, one of the things that came up, which was completely bizarre, was that subpopulations of macrophages expressed INS2. So in mice, there are, for those of you who don't know, there are two insulin genes, both functional to some degree, and I'm certainly not expert on that. But in any event, one of the things that was really quite striking was that, and only seen in obese animals, there was expression of the INS2 gene, and it was seen in both in this experiment's case in almost all the F480 low samples, and in one of the 3 F480 high. So in both the resident as well as the recruited macrophage population. So these again were sorted cells in which she did bulk transcriptional analysis, and 4 of these 12 samples, there was measurable amounts of INS2, and not just a little bit of INS2, but actually, as you can see here, KPMs that are in the thousands. But it wasn't only so this is when I saw this, I thought, oh, Larry Chen many years ago had reported that there were extra pancreatic insulin producing cells. So I went back and looked at some of his data. And this is from one of the first papers he published showing some staining of insulin by antibodies here, and then some qPCR. And in his studies, he would only see these rare insulin producing cells in obese insulin resistant cells, in obese insulin resistant animals. He saw it in the liver, he saw it in the spleen, he saw it in the bone marrow, and he saw it in the adipose tissue. He did some qPCR, and interestingly, it was not in the adipocyte fraction that he saw it, but only in the stromovascular cells. He then went on, I don't know if I have the picture here, to show subsequent MD PhD students showed that it was in macrophages that they could detect the insulin protein. What was striking, so that was interesting, and Larry had spent a fair amount of time and effort trying to initially they thought, well, maybe this might be a way of inducing insulin production in non-pancreatic cells, and imagining that it could potentially have some therapeutic benefit. But when we looked at our transcriptional data, it wasn't just INS2 that was expressed. It was also INS1, and you can see in Xiaoyuan's data here that it mimics precisely sort of the relative expression of INS1. But it was also other beta cell specific genes. So AIPP, for example, was expressed in exactly the same samples, and chromogranin B. And anyway, so we went down an entire list of beta cell genes, and sort of consulted with Mimo Chili, who has a lab next to us and is beta cell ontogeny expert. And we found PPYs, somatostatin, and a bunch other, what I would describe, I guess, as neuroendocrine genes expressed, specifically in populations of macrophages from obese animals. All total, there were about 35 genes whose expression, correlated with INS2 in all of these samples. And here they are, we sort of just cataloged them, and it seemed as though what we were seeing was ectopic promiscuous expression of these neuroendocrine genes at levels, in some cases, that were quite low, but very consistent, and sort of in comparison to the non-INS2 expressing cells, essentially it was 0 in all of them, and it was above 0 in these cases. So, and again, these are typical of neuroendocrine cells. Was this some artifact of Xiaoyuan's experiment? Did she, by accident, somehow get pancreas or intestinal cells or brain cells when she was isolating epidermal fat? The short answer is no, because Ethan had similar experiments that he was doing. We went back and looked at his mice. In his case, they weren't leptin deficient, they were high-fat fed animals. And consistent with what Larry had found years ago, we only saw the expression of insulin, and as I'll show you, these other genes, specifically in the obese animals. And again, it varied in degree, so it appeared to be a little bit stochastic, but again, was not limited just to insulin, but was actually INS2. And again, you can see quite clearly the pattern is the same. And again, it was not only INS2, but AIPP, as well as a whole bunch of other genes. So not all of the 40 or so genes that Xiaoyuan found were expressed, but a good majority, in fact, were expressed in Ethan's data as well. So is this just a weird mouse phenomenon? So what I did was go back and take a look at the Broad's transcriptional profiling data for adipose tissue in humans and looked for insulin. And here's what you do when you pull that up, and you can see that, as expected, insulin's expressed, essentially only in the pancreas. But if you look all the way over here at adipose tissue, there appears to be some person who has a little bit of insulin, again, above what you'd think were baseline. So if you look at this on a log scale, here the median expression was 2,300 transcripts per million from the pancreas. Over here, again, if you look through the database and ask query, what's the median here, it's 0. But it's not 0 for all of the samples. There's actually two samples here, as you can see, that have expression in the thousands, again, from adipose tissue. Here it's 900, here it's 200. I'm sorry, yeah, this is insulin. And again, I'm sorry, not thousands, in the hundreds. And again, if you look at those same samples, you can see that it's not just limited to insulin, but actually also glucagon. So here, that same individual who had the very high 900 transcripts per million of insulin had 300 transcripts per million of glucagon. And the person who had number 2 had 20. So it appears as if that there is promiscuous expression of these neuroendocrine genes that can occur, at least in the mice and adipose tissue macrophages. It seems to occur specifically in obese, again, at least in mice. And the data that I showed you from the Broad database provides some circumstantial evidence that it may also occur in people. So why might this be important? It's very cool, and obviously raises lots of questions. So one is, what's the molecular mechanism of this promiscuous expression? Why these specific genes? We have some data now to suggest that it's not many ATMs expressing low levels, but it's actually a few of the ATMs expressing very high levels of these genes. But the thing, as someone who's married to someone with type 1 diabetes, the immediate question was, is there immunologic consequences of this? And I'll just remind you that promiscuous expression of insulin is not new, but actually occurs and is necessary for tolerance. So promiscuous expression by the medullary thymic epithelial cells is actually critical to eliminate autoreactive T cells. And this occurs in the thymus, it's been best characterized there, but also seems to be able to occur also in lymph nodes. again, is important in eliminating autoreactive T cells. But that occurs in an environment that's specifically evolutionarily designed to generate immune tolerance. So the question, and this is just a cartoon, I realize I'm running out of time, so I'm going to sort of wrap up, but this is just a cartoon that indicates or sort of shows that. So within the medulla of the thymus, this stochastic expression of self-antigens from other tissues occurs in an environment that leads to negative selection of T cells that would be autoreactive. So the question is, what happens if there's promiscuous expression of insulin and other beta cell antigens in adipose tissue? Is this a Th1-like environment that, instead of triggering tolerance, actually is immunogenic? And so I don't have the answer. So I'm running out of time, but the basic thought is that, it's occurring in cells that in fact do express MHC2 that are capable of presenting antigen. And that our assumption is that the machinery for normal production packaging of insulin per se doesn't exist. But this may set up a situation in which the large expression of this protein and these other proteins actually leads to presentation. And as I showed you from in the single cell sequencing data, as well as other data, there are a large number of T cells in adipose tissue. And if this Th1 environment is right, it could lead to an autoimmune response. Okay, so, and I guess I will sort of leave it at that. The only thing I'll say, we don't have any direct evidence of that, but we've been intrigued enough by these data that we are now sort of pursuing this much more fully, in both terms of trying to understand the biology of this promiscuous expression, what controls it, and whether it contributes in any way to autoimmune type 1 diabetes. The last bit of data that I'll leave you with here is that the finding that obesity is in fact associated with an increased risk of type 1 diabetes, and the relative risk is on the order of 3-fold increase. So this would, again, just circumstantial evidence that would be consistent with the idea. Okay, so hopefully I've been able to convince you that there are multiple populations of macrophages in adipose tissue, and we think that we'll be able to, and that some are derived from circulating precursors, other from resident precursors, and that they play a role in lipid homeostasis, but although we've yet to be able to assign specific functions to specific subpopulations. And then I'd like to propose that there may be a way in which these cells can contribute to a loss of tolerance, at least loss of tolerance to beta cell antigens. And with that, I will end.